Okay, I am Nicole Kleinstreuer. I'm with the National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods. And I'd like to invite our panelists to come sit up at the front here. So our panelists are Nadira Abrew from Procter & Gamble, Anna Lowit from the Environmental Protection Agency's Office of Pesticide Programs, Kristi Pullen Fedinick from the National Resource Defense Council, Charles Schmitt from the National Institute of Environmental Health Sciences, and Reza Rasoulpour from Corteva Agriscience. Okay, so I think we do have the scenarios for this panel discussion in slide form. Yes, we do. Okay, so let's see. So this first scenario, so what we did here is we came up with some hypothetical scenarios that we would like our decision makers and our stakeholders to respond to, and we tried to make these, realistic, but they are, disclaimer, they are hypothetical, and so I'm going to read this one to you. So a researcher, good, Liz? Okay.
I think that's not the right version.
It is somewhat different from the version that is on the printed agenda. So I'm going to read the version to you that's on the agenda while we...
It's not, for scenario one, it's not that much different.
Right. Okay. So a team of cancer epidemiologists develop a geographic information system-based machine learning algorithm that uses comprehensive information, such as proximity to nitrogen sources, fertilizer use, hydrogeologic information, et cetera, to predict which cohort study participants are most likely to be exposed to nitrates. The model has been validated in a very small subset of homes where tap water samples were collected. The model identified almost 100% of the contaminated wells. However, 40% of the wells that the algorithm identified as contaminated were negative for nitrates in the laboratory test. So there's some false positive issues here, obviously. So what should be reported to study participants as a result of this model? How could environmental agencies use this model, considering the propensity for false positives and development on a small data set? And what if the algorithm continuously adapts with new information? What are some considerations that should be put in place to reevaluate the algorithm? So I'd like to just go through our panelists and get their initial thoughts. These scenarios were actually shared with our panelists ahead of time. So hopefully they've been thinking about it for more than the 30 seconds after I finish reading this. So Reza, do you want to start and we'll just go down the line?
Yeah, it's interesting. As I was thinking about this, the key thing that comes up is credibility that the public can have or not have with regards to scientists, and that scientific credibility with the public is a really important thing. So, although in a public health type of situation, having some degree and accepting some degree of false negatives in order to ensure that you're providing a wider protection of people makes a lot of sense. Being off by that much could lead to a lot of alarm, but also a lack of trust in science, which I think is an issue. And interestingly, I was reading this while I was reading a CNN article earlier this week about the fact that scientists have discovered there's no difference from a cholesterol health standpoint of white meat versus red meat. And I've been not eating red meat for quite a number of months because I was told that I need to be on the Mediterranean diet. And I thought, well, there it goes, right? Science, providing a counter argument to what we had before. So I don't know. I think there's a risk here, and although there's, a desire to have some sort of intervention, I think we need to be a little bit careful.
Reza took my my insightful points. I think that's right on. That when so if the first question here, I think is the really interesting one, is should the information be shared with the participants? So first and foremost, we should be erring on the side of transparency. So anything done out of the study, I would hope, would be transparent and publicly accessible. That's different than sending out a mail out to someone's house that says the prediction level of X, Y, and Z, and your house is this. Having a database where people who understand how the information was collected or generated and use it appropriately is quite different than sending it to my mother-in-law, who will have a total meltdown. And I'm sure many of you would have a similar situation. So coming at it from putting my regulator hat on, I would hope that we would not actually inform the participants of the results, but I would hope that these results would be publicly accessible, so someone could find them if they're really interested, because we do have this trust issue that we need to think about. And living in a world of Twitter and Facebook, this kind of information can be and is routinely misinterpreted and twisted in a way that people now don't trust many things. I mean, we see this all the time, that information, scientific information that people really don't understand can be cherry-picked and twisted in a matter of minutes. And so we need to be very thoughtful about how everything should be transparent. I 100% believe that, but we need to be careful about how we communicate that information.
Great. Yeah, thanks for the opportunity to comment on this. So, my read of this, thinking about this from a Safe Drinking Water Act perspective, was one of a system that has a lack of information overall about the overall drinking water quality in most American homes. And so if you have a model that can allow you to better understand a complex, situation in which we have a, relatively data poor situation, then I think that this information and these type of data can really help, one, inform where risks could exist. So, for example, if you think about, Flint, which is a poster child for lead in drinking water. So Flint hasn't shown up in the SIDWIS, the Safe Drinking Water Information System, as having an issue with lead. Right? It didn't in 2015 at the height of the Flint crisis, and it doesn't today. And so if we had additional information that could allow us to identify potentially vulnerable areas for nitrate contamination or other type of contamination, then I think that would be particularly useful. I think the disclosure of that information to the public is also helpful in terms of giving folks autonomy to be able to try to, one, determine whether or not they themselves are also at risk. So you can say that there are uncertainties associated with these particular types of data, but you may need to protect your family if you have a child that is, if you're pregnant and you're worried about blue baby syndrome, then it'd be really important for you to know whether or not you need to take personal action in order to try to protect yourself, or you can therefore be precautionary about whether or not you would want to drink water from your particular tap. And so I think that you can use these data in a way that isn't alarmist, but is informative and allows people to, again, have autonomy. And then I also think you can take this type of information and really set up accurate and more consistent testing regimes to really try to understand where it is we should test. Where are the higher probabilities of having nitrates in your water, other contaminants in your water? How do I design a testing system and testing mechanism and testing frequency? So maybe the 40% of tests that were negative in laboratory tests, they were just tested at a time in which nitrates, for example, weren't being applied because of, it was off season. There could be a number of things that could be wrong even with the laboratory test itself. And so really trying to understand what the system is, how can I improve it? How can I use these data to ultimately protect public health even more as a sort of general social safety net? But then how do I also empower individuals to be able to make decisions about themselves and their families health?
So I'm going to give a kind of schizophrenic answer. Let me first put on my hat as a data miner. Looking at that 100% yeses for true positives, 40% false positives, small data set, it sounds like the data is highly biased towards positives and that the model is picking up on that. And so my first thought is there's a methodology concern here, probably on where the data is being collected, that's giving the oversampling on the positives. So at first blush, I'd be very concerned about the data collection. Was it representative, and was the model dealing with that balance? So let me then switch on my well owner hat, because I live in a house with a well, and I've been on a well for a long time, and I have 3 kids drinking from that well. that part of me would like to see the model released in some form, because I would like to use it to test to see if I should be ordering a nitrate test. Now, I would say nitrate is pretty common. It's on every Amazon well water kit that you get. So, in terms of value, it really doesn't provide a lot of value for me. But if that was a different substance, one that's not in the common testing that you get from Amazon, then, given the inaccuracy, I would still want to know, should I shell out another $250 to pay for that test or not? So as a homeowner, I actually would like it released in some form. My caveat there is that I don't want other people running that model and seeing that my house has a well that has contamination in it. So that's a challenge there in how you would release it. I think it definitely, if it's released, it has to be released under caveats and knowing it's a research project at this stage. And with a lot of the things that Kristi said, it's a good starting off point. But it would be, I think there's some things that would have to be looked at first.
So I think my comments are similar. I think the context and the limitations of the study need to be defined when we're being transparent about it. But then maybe release the actual laboratory data to the study participants, and then kind of say, we're still working on this to, get a better outcome. So kind of balance the transparency and then also define this is what was used for this study, this is the limitation, and it's an ongoing process. So that would be kind of my input.
Okay, great. Thank you. Do any of the panelists want to respond to anything that their co-panelists have said? No. Do we have any thoughts from folks in the audience about this particular scenario? Or shall we move to our next scenario? Okay, so we'll move to scenario number 2. So scenario number 2 is that a hair care company is working on creating a new hair growth product. This hair product contains a new chemical, GrowShine. The company acquired a large database of chemical structure activity relationships for which fewer than 20% focus on neurotoxicity. A machine learning algorithm is used to predict the toxicity of GrowShine based on the information in the database. They find GrowShine may be neurotoxic. What would make you confident that this is a good application of machine learning? What are some questions you would ask about the machine learning algorithm when you know the database it's learning from is not complete? What more information would you need to determine GrowShine should be removed from the product pipeline? And what information would you need to make you confident that GrowShine was safe for use in hair products? So why don't we go the other direction this time and start with Nadira and then work our way down?
So I guess I would start with first understanding if I even need machine learning. I mean, what kind of data set do I have already? And then kind of go from there, and then moving down the questions, then try to understand what we know about this, so the database, and then the chemical space of that database versus, what is this, GrowShine? So what does GrowShine look like? Does it fall into the chemical space that we have this 20% of data that's available? If not, kind of think about, okay, can we really use that data set for this this kind of chemical? And then a holistic risk assessment approach, right? So we're just looking at one endpoint here. And so what about the other tox endpoints? And so in that context, is neurotoxicity really what's driving for this chemical, the driver for this chemical, based on what we know about these kinds of chemicals? So I would ask those kinds of questions. And then, and that final question, I mean, it's an overall risk assessment approach. Again, look at all the different endpoints for the toxicology assessment of that ingredient, and within that context, and how the in this case, the person would use it in a hair growth product, kind of understand if that's really the driver, and then will a machine learning really add to that overall bigger question, and based on that, make a decision of how much time I would spend on that kind of approach.
So I think in this case, I'm assuming that the company is doing this as a first pass to decide if they should go forward with this. So I'd have to think that the default is to take the model as right, and then the really the onus is to try to prove that the model is wrong, and if you're going to consider keeping it on. So probably the first thing I would do is ask Dr. Tropsha if he can attack and criticize his model and the data that goes into it. And if it passes that sniff test, there's a lot of things that were brought up yesterday and today about assessing models, looking at the interpretability, looking at the domain of application. Of course, you would look at all those, bring in external experts to help with that, because the cost of doing that is probably a lot less than going forward or killing a product. So it's kind of due diligence on looking at your models and understanding that the data going into them is high quality, that you bought good data, trying to trace down the provenance of that data, that your modelers are using the right techniques. So I think it's really putting it up as a challenge that the model has to be vetted and prove that it's wrong in this case. In terms of looking at the algorithm and some of these questions here, it's really, given grow shine, where does it fall in chemical space compared to where the model was built? And if it's off to the side, then it's not the right model, and we can discard that and maybe try get more data, try to build a different model. But I think it really comes down to assessing that model and seeing if it's appropriate for this particular product.
So, and thinking a bit about this, I appreciated that it's a new chemicals, not rather than an existing chemicals. I think that the comments on this would change if that were the case. So if this were a product that existed that already was on the market and had, a machine learning process that also went, that was also identified as potential for neurotoxicity. So I'd be interested in getting the rest of the panel's thoughts on that too. But assuming that it's a new chemical, I would, also follow Dr. Schmitt and say that you should assume that it's an actual signal of potential toxicity. And I think that this comes down to, again, the value that you place in the information. So do I value putting my product out and making money off of that, which is a fine value it's supposed to have, or do I value protecting the public from potential neurotoxicity? And so if I value protecting the public from potential neurotoxicity, then I would have a pretty high bar, and I'd set a pretty high bar for proving that the chemical is not neurotoxic. And so really ensuring that you are, again, understanding the value of the particular person that's going into making the decision as to whether or not you continue on with something that could be harmful to a large portion of the population, or whether or not you would move forward even with uncertainty and market a product that could be potentially harmful. So, for me, I would say that I would want to be able to go through, extensive testing to disprove that neurotoxicity. So another model couldn't do that, for example. I don't even know that high throughput in vitro studies would be sufficient for that as well. And so, if we're going to, essentially be testing this in a human population, ultimately, if we don't ensure that we have extensive testing done beforehand, then I would really want to make sure that we have all of the information we need to ensure that that signal we saw was a false one.
So in my experience, most of these models tend to be high on the false positive rate. That's in many ways, from a public health point of view, that's what they're designed to do. So if you think about it from a screening prioritization point of view, whether or not you had confidence in that 20% or not, the obvious next step is to look at it from a in vitro or high content imaging kind of point of view of doing either cell-based assays or tissue-based assays specific to neurotox, and eventually even think about a whole animal study. But I think the other piece that really needs to be brought up is that Here in the United States, we're not a hazard-based system. So simply identifying something as potentially neurotoxic or even neurotoxic for that matter, even if you have good human or whole animals testing, doesn't actually mean that it's going to not be safe in that hair product. It's a determination of the evaluation of the level at which it's safe and ensuring that the amount of whatever chemical it is far below that, assuming that there's population variability and all kinds of other things, to make that hair product safe. So I think there are a couple of things going on here that are mixing a couple of issues.
So maybe adding something different than what people have said thus far. The first thing that I would do is, if this is really early in a pipeline, there's a lot of other structurally similar molecules, right, that were created in the development of grow shine, even in those early stages. And there could be thousands, there could be tens of thousands. So actually, the first thing that I would do is I would explore the structural activity relationship around grow shine first within that same model, to see if there was some specificity associated with slightly tweaking the molecule, just as a first pass in understanding the level of specificity with regards to getting that positive hit with regards to potential neurotoxicity. The second thing would be to look at the target of grow shine, right? What is the molecular mechanism of action? Is it biologically plausible that it's causing some sort of neurotoxicity based upon what we know about how the molecule works? And I think that's an important thing just to drive what the next steps would be. Certainly before you would advance a molecule like this, or even advance a molecule that was an analog of it that didn't have a hit, you know that there's a potential concern here. Following up with the appropriate in vitro or in vivo studies to assess the endpoint, to have the confidence yourself that the molecule's not neurotoxic, or even if it was neurotoxic, that you have a appropriately adequate margin of exposure associated with that sort of product. So I think those are all important components. And it's interesting because I wonder if our answer to this question would be any different if instead of being a new hair growth product, it was a Alzheimer's drug. Or if instead of being an Alzheimer's drug, it was a drug to treat some sort of very rare form of childhood cancer for which there's no other treatment, right? So with every one of these types of decisions, there's a risk-benefit equation that needs to be put into place. And I would push back on something that Dr. Pullen Fedinick said, where there's a choice of either pushing a molecule through in order to sell the product and make money off of it, versus ensuring that you're protecting the population based on neurotoxicity. At least speaking from someone who works inside of industry, those things are not mutually exclusive, right? So they go hand in hand with one another. It's the right thing to do for the public, it's the right thing to do for the company, because you want to make sure that the products that you're putting on the market have the best human health profile possible.
Well, just to add to that really quickly, so I think that if we were going to take whether or not it's biologically plausible, in a lot of ways, I think that that's an act of hubris, right? It assumes that we understand all the mechanisms by which something could cause neurotoxicity, and we know, particularly for that endpoint, that we know very little about the universe in which, say, Parkinson's is caused, for example. And if this were a drug and not a consumer product, that also goes through a different regulatory process. It requires it to be shown to be safe and effective prior to its, use in populations, whereas products, that we're exposed to every day don't go through that same process. And so if we were to label this Grow Shine product as being maybe neurotoxic, could cause Parkinson's on the bottle itself, again, and giving folks the opportunity to say, well, we're not a hazard-based system, so, it's okay if we have a little bit of a neurotoxin within this particular product. I think that, to say that we're not particularly hazard-based and this should be risk-based, I think also ignores the, again, the potential that could exist for this chemical to be neurotoxic, given the fact that we don't necessarily understand all the biological mechanisms. We don't understand necessarily the processes that go into individual populations based both on genetics, based on, say, social determinants of health and other non-chemical stressors. And so, I think, again, that the bar would have to be extraordinarily high, at least in my opinion, to allow that, a neurotoxic chemical to exist in a consumer product, given that the regulatory structures would allow it to exist even if there were a risk associated with it, to allow that to occur in this particular product. And so the regulatory mechanisms are different, even for pesticides and for, again, consumer products. So it's a false comparison in a lot of ways.
So I'm curious if anyone in the audience has opinions on this discussion. If you do, please go to the microphone. In the meantime, while Alex is going to the microphone, I'd like to put something out there that occurs to me when you're listening to this conversation, is that, no model is perfect, right? There's the potential for false positives. And so I wonder if this concept of a stigma immediately becoming associated with a prediction, and how that prediction, is associated with this chemical or this product, how do you sort of test your way out of that? Can you ever shake that stigma? So if you have a prediction of neurotoxicity for a particular chemical, and you can acknowledge that that prediction may be wrong, are you ever able to get to a place of certainty that that chemical is safe and shake that initial prediction of neurotoxicity, no matter how deeply you dig into the model, no matter how much testing you do on that particular chemical? Where is that balance? Because if not, then that's a huge deterrent to even being able to use QSAR models in regulatory decision making.
That's a good question. And I think part of it depends on the endpoint. And part of it depends on how early or late within the entire discovery process you run this type of model. So there's a reason why we run these types of models very, very early on within the discovery pipeline within our organization, because it is actually a good way to help you narrow down the structural activity relationship space, right? So running this when you've figured out exactly what's the analog that you are betting your hat on, that's too late, to be honest with you. So the utility of these types of tools actually is much earlier on, as you're trying to narrow down the chemical space from hundreds of thousands of molecules to slightly fewer molecules, because then it's a little bit okay to throw out the baby with the bathwater at that point, because you have a lot more flexibility within the SAR to explore the molecule. As you get later on down the stages, then the onus is high to provide as much information both internally and externally for the molecule not to be neurotoxic. And I think it also depends on the complexity of the endpoint.
Okay, well, Anna, go ahead, and then we'll go to the audience.
So Nicole, you and I are good friends, so I'll push back on your question as actually being flawed, that in fact models like this should not be used to label something, that it's a misuse of that model to provide that label from the beginning, that it's a screening tool for which you would then use many other tools to provide information on what the appropriate decision-making in the context would be. That's...
Okay, Alex?
Okay, so I think it was intended to be a trick question that we took way too seriously. And the reason I'm saying this is that from statistical viewpoint, a model developed on a large data set with 70% accuracy only means that when applied to a large data set, you expect to predict 70% of this data set accurately. It does not translate into prediction accuracy for a single molecule. And a well-known phenomenon of this nature in statistical literature is that when a student answers the question of what is LD50, there are two statistically correct answers. One, it's a dose that kills half of the animals. Second is the dose that half kills every animal. I'm just using this as an example of explaining why I think it's a trick question. So I think when it comes to issues, when it comes to answering this question, the types of models that we develop do not translate from big data statistics to single data point prediction. And that's a very important area of statistical modeling that should be addressed. And second, when it comes to single chemical, it should, I think, forget the modeling, it has to be tested in regulatory proof, reproducible, yada, yada, yada tests. So that would be my answer. Thank you.
Okay.
Hi, my name is Tom Barnum. I'm at USAID. I'm listening to this and I hear a very nuanced discussion, which is great. But often decision makers want an answer. They don't want a discussion. So if you had to run this model and the CEO of the hair company said, what do you think? Like, what's your recommendation? How would you approach that particular question? Because it's now changed from the nuance, well, we can do this, we can do that, or whatever, to something where now you need to go yes or no. And keep in mind, I mean, Austin, you know this, the people that you're talking to aren't dumb. They may be non-technical, but they're very intelligent in the things that they know well. So I was just kind of curious what some of your thoughts on that might be.
Yeah, so I'll give you, I mean, it's not this example, it's not Grosjean, it's not neurotoxicity, but this happens all the time, right? This happens all the time. Because you run screens early on in order to the hope to catch something, to prioritize work. So the answer is that it's a yellow flag, it's a potential concern. If it's not early enough to work your way around it through a different structure, then what you would do is you would start off by figuring out what are the right, more definitive studies that you would pull earlier, right? Because sometimes there are some studies that you wouldn't do 4 years later. And the reason why you run these sort of screens is you identify something that could be a concern. You take that study that you wouldn't run until 3 or 4 years later, and you'd run it now. And it may not even be a regulatory study. It may be a study that is tailored in order to try to answer the question as best as possible, to provide you confidence on whether or not you should continue to invest in that product and invest in that molecule.
Hi, Marianne Tikimurtoglu, Columbia University. I think my comment is both on this and the previous scenario. I'm not familiar, disclaimer, with structure-activity relationships, but I think both of these could be reshaped as an exposure measurement error or some sort of bias that we have seen in other settings, not in machine learning or artificial intelligence, with more traditional or conventional approaches. And I think it's very important, not necessarily for regulatory action, but because of the hype, I think we tend to ignore that these are biases that we know exist and that we should be developing methods that can be attached to machine learning to correct this. I think that machine learning, because of the adaptivity of these approaches, can actually help us do that. The more we fit it, the more it would correct. This will not help this situation right now. We need an answer now, of course. But I think these are issues that should be discussed, and including, I come from an epi field, but including the biases that we know and how we can correct for these biases using these methods should adaptively get us closer to the correct answer. Especially for this, we have now the opportunity to integrate very different data sources. So we will be able eventually to include tox data from my studies in the same analysis as the structural activity relationships, potentially even eventually epi data as well. So I think as long as we recognize that and are transparent and work on that, in a few years we'll be at a much better place.
Would our panelists like to respond to that?
Well, so I don't know if this came up exactly in what you were saying. Maybe it just popped up in my mind as you were talking. So I apologize if it doesn't address it directly. But one of the comments that Dr. Lowit just made, saying that these were, screening tools and we couldn't use them necessarily for identifying a potential harm. And so if we're thinking about biases and, thinking about this as an exposure, so if this were a new chemical application, so if this were going through a TSCA process and they were applying for a new chemical status, and this was the only information that you had available to you, I would say this isn't a screen, it's an indication of harm. So it's not a chemical that should be allowed under, say, TSCA as a new chemical. And so I think that we have to think not only about, again, some of the biases that are associated with this, but also the decision context in which you would be using this type of information. And so if this is all that you have to go on, for example, so if you're a company developing it, you have different, processes available to you to try to ensure that that particular chemical is safe or not safe. But I think that if you have a lack of information completely, and this is what you have available to you, then the signal would be to say that this is something that's harmful to the public. So I think we need to move beyond thinking about them simply as screens, but also indicative of actual potential toxic or actual toxicity. So it's more than just a screening tool. It really depends on the context in which it's used.
And I think what you were also referring to is that there's an onus upon all of us to make sure that as we're working with these models that we continue to improve them, right? Because fundamentally all models are wrong, but some of them are useful. And I agree with the previous example, and with this example, it's important to anchor the data, particularly the previous example, with why is reality or what we're observing not matching the model? And is it because we're looking at the wrong time and maybe the model actually has some sort of sensitivity that we're missing? Or is it that the model is making some sort of false assumptions, right? So continually improving that is helpful. I think one of the challenges there is there are so many different models. We don't do this in enough of an open source kind of way, although I'm encouraged that that's going to increase in the future. And we also don't do it enough in a transparent way, but I'm also encouraged that that's going to increase in the future.
Hi. So I actually sort of wanted to jump in, I guess, to say what I would have said at the very close of the last panel in response to the question about how to make visible the values of the modeler. So the subfield of computer science that I come from is called Fairness, Accountability, and Transparency. And there's a bunch of people in that community who have been thinking about questions of modeling as it applies to society. So high stakes decisions about people, right, which I think is also really what we're talking about here. And part of the set of, I don't want to call them solutions, right, but the set of approaches to trying to address some of these questions that you've been raising, that people in that community have been taking that might apply here as well, are creating documents that should be associated with each data set or each model that make very transparent what the aims were in creating that model. So for example, these points that you guys are raising about how the purpose might actually be to try to have more false positives so that you don't have as many false negatives, right? Listing out all of that type of information and what's being referred to by a bunch of different names, but some of the names are model cards or data nutrition sheets, these types of things that make it very clear, this data was collected from this particular subset of the population at this time in the following way, right? Here's a card that goes with my data set at all times that explains all of these things, right? This model was trained with the purpose of having a low false negative rate, and it was done in the following way, and you could replicate that model by doing this thing, and here's the specific subset of the data that it was trained on, and so on, right? Here are the evaluation metrics that I looked at, here's the ways that I tried to make sure that the model was not biased, here's the results that I found, right? It's a full sort of evaluation sheet. That is, the idea is it's supposed to accompany the model so that when you're faced with something like this, you can say, okay, it's not just that it was some model that found that it might be neurotoxic, but that model I know was tuned to, specifically provide for a screening mechanism. And I know that like the designer of the model, when they made that, their goal was that it should be used to, filter out, right? Because maybe it wasn't in this scenario, right? But if you knew that, that would give you some added confidence in how you responded.
Would any of our panelists like to respond to that?
I don't have an answer to that one, but I actually did want to go back to what Nicole had said earlier, because it's been kind of bugging me for while we've been sitting here about the stickiness of what the model says about a chemical, and does that stickiness kind of perpetuate? And I understand on the regulatory side that in the industry side that there's kind of gates in place to keep that from happening. From the side of being in a group that is actually looking at making data more open, making models more open, making information more open, that's actually, kind of a concerning thought about how the system corrects that overall. And I'm not sure I see any barriers or mechanisms other than someone publishing a research paper saying that model sucks as a control on that information getting out. So I'm not raising it as I know the answer, but I do think it's a point that needs probably more attention, especially because we do know that industry and data science on the information side are fairly promiscuous about using information for their advantage. And so I think that's a concern that shouldn't be tabled very quickly.
Yeah, I mean, I agree. That's why I posed the question. I mean, think about the example of autism and vaccines. That's something that's been totally discredited. The one study that was published was retracted. It was, it's known in the scientific community to be completely false, and yet there is a continual increase in the number of unvaccinated people in the population. And so we have recurrence of outbreaks of diseases that, should have been eradicated, right? And so my question was not really in the context of what a regulatory agency would do to dismiss that, but what citizen scientists, so to speak, and the public do with information once it's transparently shared with them, even if it is disproven sort of along the course of additional testing. And so, how do we handle things like that?
Yeah, I think that's great. I think the flip side of that, though, too, is we have chemicals like formaldehyde, right, which we have an overabundance of information. We know that there are, carcinogenic or, cancer endpoints associated with exposure to formaldehyde, yet it's everywhere. Right. And so I think that we need to grapple with both, say, the false positive situation for, say, new chemicals, or if you have a model that shows that every chemical in the world is toxic, you don't want everyone running around thinking that they're completely unsafe. But at the same time, I think that we exist in this system in which the overabundance of data still doesn't remove toxic chemicals from the market. And so how do you think about shifting the value structure and really trying to understand, again, whether or not we assume that all chemicals that we're exposed to and the levels that we're exposed to are safe, whether or not we assume that they're all bad and need to be proven safe, what the levels are for the different ways in which, if we're structuring those value systems in different ways, what would be the level of evidence required in order to go in the other, more say, protective direction. And so I think it's not just a vaccine situation, right? I think it's also a situation in which we have asbestos. We have a number of chemicals that, again, we know that have extraordinary amounts of toxicity, but aren't regulated in the way that they should be, right? Because of the system we have in place that really protects chemicals over people. And so, I think it's really just a tightrope that we have to walk and grapple with on both sides of the equation.
So,
Identify yourself.
Oh, hi. Sorry. This is John Wamba from the Environmental Protection Agency. So, I'm not on the regulatory side, but I'm sitting here imagining myself working at this company, and this question comes up, and the thing that really sticks out and bugs me more than anything else is that the company acquired this database of QSARs, which, I guess you look gift QSARs in the mouth is kind of how I look at it. And what I would want to know to have confidence in this is if the compound's important and the CEO says generate some more data, can I use that QSAR to incorporate the new data? And I mean, these are things that are said there. Just the knowledge of whether or not that is a possibility honestly makes me more comfortable with the model. I don't know why, but that's just a weird, and I'm completely serious. Like the idea that it's a model that is built such that I understand it well enough that I could get data and improve the model. If I can't get data and improve the model, if that's not even an option there, I don't trust the model. And so I don't know why, but that's just my gut feeling on it.
Thank you, John. Okay, so we have less than 5 minutes left, and I just want to ask another series of questions around the concept of using artificial intelligence or machine learning in decision making. So we have a number of additional questions that I'm sort of going to combine into one, which is, what are our panelists' thoughts about, what are the best practices for using models in decision making? what should data scientists be prioritizing when they're building models that are intended to be used for public health decisions, and what types of policies or guidelines should there be to help data scientists in guiding them to better create models that will be more useful for public health decision making.
So I'll start so we can get answers wrapped up. One of the things that struck me during this meeting, especially yesterday, was kind of two things. One is ensemble models are good. We should use them. And second was interpretability is good. We should try for that. And those two are kind of hard to put together because a model by itself is hard to interpret. A bag of models is even harder to interpret. I think one of the things that's come out and that's encouraging is that we've seen a number of ways to interpret machine learning algorithms. The variable feature permutation test, the regions of fit for models, even with the neural networks, the ability to look at the decision boundaries that it's drawing in middle layers. So there are techniques that are coming out. I think the challenge for the community that would help in moving this forward is getting to the point where everyone is comfortable with the approaches, so that when you build a model and you communicate it, the decision makers understand how you interpret that model and value it. Because I give you, just for instance, if I present a regression result to a clinician, I know what questions I'm going to be asked about the model and how to interpret. But if I present a random farce, they typically won't know what to ask me about that. And I think that getting that communication about how to look at machine learning models in decision making is important.
I can go next. So I think you have to define the context. So I think different industries could use this different ways. So speaking from where I am, we don't really have a screening process. We're way down at the very close to a product going out to the consumer, and that's where we're asking the question. So we need to be very specific and very defined about what the model can and cannot do, and then kind of use the information from the model in at the end innovative evidence kind of assessment. So it's in my case, it's never going to give me the complete answer, but it could help me in an area where I still have some questions. It could point me in the direction of, okay, you need to focus on this area a little more, using currently used methods. This is probably the area you can focus on for this specific question. So I would say very cautiously and being very specific about the boundaries of the model is how I would apply it.
I'll go next. So a couple of thoughts come to mind. First, context is vital. One model can be very useful for one context, but be completely useless in another one. So understanding the decision-making context is vital. So whether you're developing a QSAR, you're using it, you're making decisions on it, you have to understand not only the QSAR itself, but the context under which it's built and the context under which your decision is making, and make sure there's some reasonable overlap between them. Something else that really hasn't been brought up at all is that, so I deal with decision makers all the time, have for a very long time, both at the career staff level, but also at the political level. And at least in my experience, there's a lot of misunderstanding about decision makers, that they're simpletons, that they want yes or no answers. In fact, in my experience, that's the furthest from the truth. That in fact they don't want yes or no answer. They want to understand the boundaries of the information that you're providing them, what all the alternative approaches, alternative options may have been, or maybe in the future. And so, decision making is a very complex set of things, and it's extremely rare, at least in my in my experience, for those to come off of a single piece of information. In fact, decisions are made from lots of information from diverse sources, not only science, but other sources also. And so to keep all of that in mind, so when we pull out just these questions on the newer tools of thinking about big data and how it's used, it's going to be a rare instance when that is the only piece of information that's used in some kind of decision, whether it's a small decision for a particular chemical or particular use or use site, or it's a big decision of we're going to completely remove something from the market, or we're going to bring something brand new that no one's ever seen before. Those are big decisions. None of those are made on individual pieces of information.
I couldn't say it better myself. I mean, it's the tools need to be context dependent. And I think making sure that we have, and for the people who are making the algorithms, the time to sit with the decision makers with the context that those decisions are made, to understand exactly how those decisions are made and what are the trade-offs that need to go into those decisions will make the algorithms better. We talked yesterday, Gary brought up the fact that, algorithm and computational scientists are often magicians. We talked about David Copperfield and David Blaine. And I think the easiest way to do this is not to turn us into David Copperfield or David Blaine, but bring them with us and have the conversation together so we can see the magic trick, but then also understand how it was made.
Yeah, and I would say, I'd concur that decision context is really important. And again, just getting back to the idea of values, I think that we have to weight information in different ways based upon the type of decision that we're making. And so if we're under a regulatory or legal context to protect public health, to protect the health of ecosystems, to protect vulnerable populations in particular, then we need to weight information of harm in a way that's different than we would weight information about, a lack of harm. And so even if I have, a multiple or a number of things that I'm looking at, again, thinking about a new chemicals context, the indication that there could be harm would be something that that we should think about weighting differently if the goal of the particular decision is to protect public health. And so we not only need to think about the decision, but also think about the goals and what it is that we're trying to achieve with that particular decision.
Okay, well, I would like to thank all of our panelists for a really tremendous discussion. And now I'd like to invite Gary to come up and close the meeting for us.